All right, so I just started it. Okay, so again, we're just starting with quickly looking at or revisiting the what we've learned so far for the last 7 lectures or so, and those were about enzymes as well as metabolism. So first, we're going to go over the enzymes and the catalytic reactions. So first, we talked about many things. We've actually started with the some thermodynamics and their basic concepts that are relevant for the catalytic reactions and so on. One of the important things is that we defined the delta G double dagger as well as delta G double dagger. So you should be able to distinguish these changes and understand what they mean. Basically, that's all about defining the free energy of activation, right? Which is different from just the free energy, delta G versus delta G double dagger, and catalyzed reaction versus uncatalyzed reaction. Okay, let's move on. And I mean, even if I just skip saying something, that doesn't mean that they're not important. So you should, study all those things that we've learned and discussed so far. But again, these are the important things. And then we talked about serine proteases as a good examples for those, enzymes that boost the catalytic reactions. And we basically talked about these 3 different clans, major clans, and focused on this chymotrypsin-like protease, serine proteases mostly. And we also talked about the 3 different serine proteases, chymotrypsin, trypsin, elastase, and so on. And they have preference basically for their enzyme or the substrates. And you should understand what kind of features that confers these kind of preference, like is that rather charged or the shape of the cleft or things like that. And we already talked about that, right? So, and we also talked about the zymogens, what they are, as well as how they are processed for activation and so on. And also cofactors too, and they have different types, as you can see, the metal ions, coenzymes, and so on, prosthetic groups, and so on. So you should be able to tell the differences between them. And also we talked about that, how coenzyme is used for activating enzyme, right here. And then we talked about the different types of coenzymes, metabolites coenzymes, as well as vitamin-derived coenzymes and their differences and so on. And vitamin-derived coenzymes have 2 subcategories here too, right? Water-soluble or insoluble. And then we moved on to the catalytic mechanisms, how we can actually show these mechanisms, the steps of catalysis, and what kind of things we use, like 2 dots mean something, arrows meaning something, arrowhead, and so on. And we talked, quite a lot about different types of catalysis. There are different types, majorly 5 different types. And of course, I mean, that doesn't mean that one type of catalysis happens exclusively. You can always get the 2 of them or multiple of them happening at the same time, but still should be able to tell the critical differences between them and basically their definition that you need to be aware. So acid, base, covalent, and so on, here. The proximity and orientation effect is important too. And then we, went over the Michaelis-Menten equation or the assumptions, as well as the Briggs-Haldane additions to explain this steady state of the ES concentration by adding the couple more assumptions there. So again, this is an important concept to understand the how enzymatic reactions happen and how they are regulated. And also we talked about the different quantities that are used to explain these enzymatic kinetics. So these 3 terms are really important: Km, Michaelis constant, Kcat, turnover number, and the Vmax, which is the maximum rate of the reaction that is achieved by the enzyme or catalysis. Again, you should know the what they mean, how you can derive these values, what are the relationships between them, and so on. And again, Kcat over Km, the ratio is important for as they are basically this ratio is the measure of catalytic efficiency or specificity of an enzyme. So to better understand how the kinetics is regulated or explained for the catalytic reaction, we talked about the rate law, and we used either just one substrate or two substrates and so on, and we could basically tell that the order of those reactions. And based on the order of the reactions, you apply different type of rate law there, basically. So you should be able to tell whether this is first order or second order or so, and what they mean too. And then very importantly, we talked about the Michaelis-Menten equation, where you can get it like this. And that basically explains the relationship between the Vmax and Km. And you can determine the Km value, Michaelis-Menten Michaelis constant, and the Vmax by plotting V, the rate of the reaction, and the concentration of the substrate here, which shows this kind of curve in the hyperbolic curve. And you can basically convert that relationship to the linear line or the relationship here using Lineweaver-Burk plot or double reciprocal plot. And here's the equation for that. And of course, this is really important because it shows a lot, it actually tells a lot of things by looking at the slope itself or the x or the y intercepts too. And also basically, this shows the same relationship between Km and Vmax, and you can empirically get them, right, by measuring the substrate concentration and its rate with the different values for the substration, and can just simply measure the velocity to get those relationships and calculate these values. And we also talked about the inhibition for the enzymatic reactions. So irreversible one is pretty simple, where it's going to be permanently inactivated. And we basically spent quite a bit of time to explain how the reversible inhibition works. And there are three main different types, which are really important to understand. Also, should be able to distinguish them. Competitive, uncompetitive, non-competitive inhibitions. And not just simply, going over the definition of them. We actually try to explain their mechanisms by, using the kinetics there of the enzymes. So for that, we use the Lineweaver-Burk plot. And this is an example of a competitive inhibitor where you can see the Lineweaver-Burk plot changes. And it has a peculiar change there. Here, as you can see, the y-intercept is constant for competitive inhibitor, but only the slope changes because the parent Km increases, but the Vmax stays the same for that case. And we also, have driven the equations for that to explain how it actually works. And this is a Lineweaver-Burk plot for uncompetitive inhibitor. And you can see the change there that is different from the competitive inhibitor. And this is non-competitive inhibitor too, where the change is obviously different. And E and S or EI versus ESI complex, which is for the K prime I here. So again, revisit these and make sure you understand this, how it works. And this is a nice table where it shows all the different quantities that are involved for their changes for depending on the different types of inhibitions. Okay, so that is pretty much everything that we've learned during the catalysis and the enzymes. Do you guys have any metabolism and also different kinds of metabolism, which is catabolism and anabolism. And we also talked a lot about the different types of regulations for enzymes that are involved in this catalytic pathway or the metabolic pathway. Enzyme availability changes is a pretty simple one where you can just simply change the amount of mRNAs or the proteins for the enzymes. But the changing or regulating the enzyme activity is more like a sophisticated or versatile method for the cells to regulate the enzymatic reactions or their activities. So there were different types of them, including this allosteric enzymes, as well as reversible, irreversible covalent modifications, and so on. If I can go forward. So this is just one example of that, regulation on the enzymatic activity, where it uses allostery. And again, I mean, we talked about this multiple times, so you should know what they mean, what it means, and how they are regulated, how they are used, basically. It is not about allosteric activator binding to an active site, but it's a different site other than active site to leads to the conformational changes, changing the activity of the active site or the shape of the active site to either inhibit or activate or promote the binding of a substrate to the enzymes active site. And we also talked at this special property of allostery, where it can have a positive cooperativity if it has the multi-subunit if it is a multi-subunit homotropic enzyme. So an example was that we talked about hemoglobin, about that, where it changes in this substrate saturation curve from hyperbolic to sigmoidal. And inhibitor basically works in a similar way here, changing the curve of this substrate saturation. And then, of course, I mean, throughout this course, we've used a lot of the different functional groups, so you should be able to, identify these functional groups. I'm not going to ask about just showing you one structure and just ask what this functional group is, just that simple thing. But at least I want you to distinguish, identify a certain important functional group, how it is used for certain biochemical reactions. So for you to do so, you need to be able to, basically tell what these functional groups are and what kind of properties they have. So it doesn't simply include this simple functional groups, but also includes these kind of bulky groups, functional groups that are widely used, as you can see here, for the ATP or the nucleic acid and so on. And we talked about the oxidative states or the reduction too here, and their definitions, and the oxidation of carbon too, where basically when carbon has a pairing or the covalent bonding with the hydrogen, that means that's less oxidated as opposed to the carbon with the oxygen, meaning that it's more oxidated. So oxidative states goes up for that. And of course, we talked about, redox reaction, oxidation reduction reaction a lot. So at this point, I just assume that you guys are well aware of this concept. But again, make sure you understand fully how it works. This is basically two half reactions. One's oxidated, the other's reduced, and one's working as a reducing agent, which is an oxidized form. And the other one works as an oxidizing agent, which is a reduced form of an chemical or whatever the compound. And it can be also called dehydrogenation when the hydrogen atom is actually leaving from the one compound. And after that, we basically spent like at least 2 lectures or 3 to discuss all these different steps of glycolysis, TCA cycle or citric acid cycle, as well as electron transport chain. It's all about breaking down the macromolecule, basically, glucose. We started with the glucose, and then breaking it down to generate energies to be used for the cellular system. So the first thing was that we talked about was the glycolysis. So this is an overall equation of that. And as you can see, starting from the glucose to the triose phosphate, which is GAP and DHAP. So you have two molecules here, and then there are ATP that are used, certain number of ATP, and also after that, basically there are two legs, which is one started from GAP and the other from the DHAP, but DHAP has to be converted to GAP basically for the further processes, and you know that how many NADH and ATP were generated there. So here's a quick glycolysis summary. Again, overall equations here. There are important control points there, which are really important because that's the step where you can, the cell can tell that whether it should go forward and process it further of these molecules to generate energy, or just stop it here because you already have enough energy there. So you should understand, you should know these are all important control points, and also committing step here where the phosphofructokinase is involved. And after glycolysis, in the end, you get the pyruvate, and this pyruvate gets to the three different vates here. One's fed into the citric acid cycle or TCA cycle, or it can go through the fermentation. And depending on which species that you're looking at, it can be go either to the homolactic fermentation to generate lactate, like mussels or so, or it can go through the alcoholic fermentation in yeast to generate ethanol and carbon dioxide. And here's a TCA cycle that we talked about. So I'm basically not gonna go over all these steps, but these steps are all important. But that doesn't mean that you have to memorize all of them. But at least for the critical steps that you're seeing, like some peculiar steps like generating NADH or GTP and so on, are definitely important. So here's a summary of a TCA cycle. The reactions here, basically TCA and the overall ones there too. And again, the important thing would be in the end, how many molecules that you get and what they're used for. And here's the balance, and that in the end, the last lecture we talked about the oxidative phosphorylation, where NADH is used to generate 2.5 ATP, FADH2 will generate only 1.5 ATP. So those are the differences there. But in the end, after the glycolysis and the TCA cycle, you're going to get a lot of ATP. And oxidative phosphorylation is important, as well as a substrate level phosphorylation too. So again, remember, I spent quite a bit of time to talk about the differences between them. So that is important, which is basically about the involvement of oxygen and the electron transport chain. And we talked about the reduction potential E, as well as standard reduction potential E naught and delta E naught too, right? So what that means, and whether it is positive or negative, and that those relationship for to have reactions for redox reaction, where the delta E have to be applied, and what that means, in terms of the spontaneity of the reaction, and the potential energy that is generated, and things so and so forth, are important. And here's the overall path of electrons that goes through, the electrons goes through the electron transport chain. So there are two paths here, and we already talked about that. But only the difference is the beginning stage, where whether it goes through the complex 1 or complex 2. and it will actually generate different results, right? And also how this ATP is powered. So it's all about using this chemoosmotic pressure or the state where you make the proton gradient across the inner membrane there. And it'll be utilized for generating ATP, for driving the ATP synthase. So we talked about the ATP synthase. I didn't have enough time to go over that because it was sort of towards the end of the last lecture. But I talked about F0 and F1 complex there. And more importantly, the mechanism. So it is quite important to understand how it is actually working, what kind of states or the status it can have, like open, loose, tight binding, and how they're changing or alternating, basically here. And there are inhibitors here too, right? For that, that affects only the ATP synthase as well as some other inhibitors affecting the transport, electron transport chain too. And this is a sort of the last one that shows the pretty much everything for metabolism. So that's it. Any questions here? And as you might have noticed, we haven't talked anything about anabolism here, but definitely, I mean, if you're gonna take the biochemistry 2, that's gonna be discussed. And given the time, we couldn't actually do that, but that is also a really important step. But we're applying the pretty much the opposite pathway of the catabolism to explain anabolism. Okay, so if there's no questions, yep. So I think the question was about whether you need to understand or should be able to drive all the equation and their steps. You should be able to, whether you need to understand all the steps or you can just understand the last step and can be applied. So it's more like the latter one. So as long as you understand the relationship, so it doesn't really have to be equations per se. You don't need to just simply memorize those equations, but as long as you understand the what they mean, in the end, it's all about the making the relationship between different states, right? So as long as you understand those relationships, you should be good. Okay, so we're gonna quickly go over the answers of that to see that whether you have any questions or so. Some of the questions are pretty obvious. And again, I mean, this is your final exam will be in the similar format as this, with a similar kind of level of difficulty too. So the first one, what's the answer? It's D, and hopefully this is pretty obvious to you guys. Enzymes, of course, at the beginning we talked about the pH, that the enzymatic reaction can go up till the physiological range of pH, which is around 7, and then diminishes after that. What about number 2? It's A. So again, I mean, it's something you need to actually understand the differences between all different kinds of inhibitions. And number 3, it's D. So this is from Michaelis-Menten equation, right? Where it relates the Km and Vmax. And number 4, it's E. And again, as long as you understand the differences between these, different type of catalysis, like acid-base, or allosteric, or covalent catalysis, or orientation and the proximity and orientation, you should be good. Number 5. Everybody's quiet, but I'm gonna just go ahead, and that's D. And again, cofactors, you should understand the the cofactor, what they are used for, and the what type of cofactors exist. But it's all about the supporting the other enzyme to make it into the holoenzyme for their activity. And number 6, it's D. Any questions on that? If it is clear, that's fine, because I thought it could be a tricky question, to be honest. But it's okay, All right, what about number 7? It's A. So again, coenzyme, you should know all the different components that are involved in the electron transport chain. And it's a pretty simple fact. Number 8, that's D. And we talked about it, right? ATP, flippase and so on. And number 9, it's A. Again, this is a simple fact that, you should know. Again, this is an important, step where it synthesizes the ATP. So it's not just one step here, right? But there are different steps. What about number 10? Substrate level phosphorylation. Anyone? That's A. Any questions here? It's just a definition. Some of you look confused, so I'm not sure actually that's a actual confusion or that's more like I don't know. I mean, you know the answer kind of. I don't know, but just raise your hand or let me know if you have any questions. Number 11. That's C. Any questions here? Again, this is not a complicated thing, as long as you read, watched or attended the lecture and studied the different complexes in the electron transport chain, you should be able to answer this. Number 12. Anyone? No. Is B. Any questions? Is it too obvious, or I actually, posted this answers like yesterday or so. So many of you guys, I assume, already looked at the answers and kind of checked it. Number 13, curved arrows, electron pairs. Number 14. Let's B. Does everybody understand this? No? Number 14? Yeah. So this is another kind of, a trick question, sort of, not really trick, but can be tricky. So here it asks the velocity. So this is about the rate of the reaction, right? And when you recall the equation for the rate of the reaction, that's V equals K, which is the rate constant of a reaction times substrate concentration, or any kinds of reactant concentration. Does that make sense? So there's no gas constant or temperature or anything involved there. Actually, it's already incorporated into the K, which is a rate constant. So it's about simply getting the product of reactant concentration, which is 1 in molar, and the rate constant 2. So 1 times 2. That's all. Does that make sense? Hopefully you don't need a calculator for this. So number 15. It's A. Again, it's the same thing I've already asked earlier, right? It's about the Michaelis-Menten equation. So, and this means that the Michaelis-Menten equation is important, especially for making relationship between Km and Vmax. Number 16. So it's E. And we talked about the functional groups that are important, right? So this is a case where you need to be able to tell what kind of functional group is there and what its implication in terms of a biological, reactions. Number 17, it's B. It's all about, carbon hydrogen bonding versus carbon oxygen bonding, and their numbers. Number 18. Hopefully this is easy enough for you. It's just looking at the delta G naught prime value, and that's it. Number 19. Phosphoprotokinase is important for checkpoint, right? And also it has the allosteric regulation there. The answer is C. So it's rather ATP, citrate, which are the working as inhibitor or activators. Number 20, it's B. Again, simple fact. Number 21, it's C. And we talked about this actually. So as long as you understand. Are you guys good? No? One good thing is that to revisit our lectures, last lectures, and just either watch the recordings or the looking at this presentation slides. But I think I've just explained this verbally when I was, in the lecture about I forgot, but somewhere in the enzymes, where there's a position changes of a hydroxyl group from C3 to C2, and that was making the either tertiary alcohol, which has carbon with the 3 different carbon groups, changing into the secondary one, which is attached to the 2 carbon groups, but the other one is different group that I don't remember anymore. So number 22, A. Hopefully it's an easy question. Number 23, it's C. Again, it's all about the distinguishing between substrate level phosphorylation and oxidative phosphorylation. Number 24, it's B. Again, as long as you understand the glycolysis, should be able to tell, answer this question. Number 25, question. Okay, let me. Yes, yeah. So glycolytic, yeah, that is correct. So I see what you're saying now, because it says glycolytic synthesis of ATP. So it might be a little confusing. Is it about synthesizing glucose or something like that? Oh, no. So, gotcha. So glycolytic pathway, basically, which is glycolysis, is all about making the pyruvate from glucose, and that's it. TCA cycle is not part of glycolysis. All right, so again, we talked about this, number 25, and number 26, it's a similar thing, right? As we've already answered. Let's see. Again, as long as you can tell allosteric and what are the activators and inhibitors. Number 27. It's C. Number 28. It's B. Does that make sense to everyone? No? So how can anyone explain why the right answer is B? Anyone? So what is K cat over Km? What is it? Exactly. That's an efficiency or specificity of an enzyme, right? So an enzyme near maximum efficiency. So you're talking about efficiency. So it should be K cat over Km. And if that is near 10 to the 8th, that means that's near the diffusion rate. Diffusion is something happens without any blocks, right? It just happens by itself, by a Brownian motion. So that means it's close to that, just a free diffusion. So that means enzymes, as soon as it meets the substrate, it's going to process it immediately without any kind of blockage or slowdown. So that's why the B is the right answer. Question? Diffusion rate is about 10 to the 8, 10 to the 9 molar square inverse. So that's something, yeah. I mean, I think I've said that quite a few times, but it wasn't maybe too clear, when I was talking about it. So whenever you see 10 to the 8 to 10 to the 9th, that means that's close to the optimal, like the rate that you can get. Number 29, it's about different types of inhibition, right? It's B. And does that make sense to everyone? It's about, when it changes only the keep the same x-intercept, it's the non-competitive inhibition, right? So again, as long as you understand different types of inhibitions and the what their features are, what their consequences are, you're going to be good, and we'll be able to answer that. Yeah. Yeah. So here the x-intercept is constant, as opposed to it changes other things, right? So it changes the slope and change the Vmax to here. So in that case, that's going to be non-competitive inhibitor. So you can actually go back to that. I can jump that, but whenever you get to that inhibition part where it explains the different features of the Lineweaver-Burke plot changes, depending on different types of inhibition, you can actually tell that. So. And lastly, number 30. That's C. And again, as long as you know the overall equation, you should be able to answer this. So I think that's it. Again, whenever you have any questions, let me know. Okay, so it's all done. So I'll get to you. Again, good luck. The exam is on the 14th at 8 a.m. It's from 8 a.m. I think it's here. So don't forget, you should come here. It's quite early, but and you have 2 hours. So you can come late, that's fine. But whenever the first student leaves, then the new students cannot come in, which makes sense, right? So make sure you're coming in quite early, So I'll answer to that question first, and then we'll get to you. Okay. Oh, so I think I said, in the last lecture, but there are a total of 30 questions for the final, but there are 4 bonus questions. So you can get up to, is it 113.3333 points. So there's another like 3.33, bonus on top of all other, bonuses. So good luck.